In this second part of the video, we are discussing the different types of digestive enzymes that are used to digest the different types of substrates. So here, a particular enzyme, it can bring about one type of chemical reaction. That means enzymes are specific in nature. So as we have already discussed in the previous part of this video, that enzymes are specific for specific reactions. That one enzyme that is catalyzing a single reaction is meant for that particular reaction. No other enzyme can act on that particular first reaction. So we can say that enzymes are specific in nature. So for example, the enzymes that break down proteins, they cannot break down starch or fats. So that means we need all the types of enzymes for breakdowning of the carbohydrates, proteins and fats. Now there are some classes of enzymes that act on different kind of substrates. Now here we have some classes which we are taking up. So first class is the amylase. So amylases, they are acting on starches and they result in the formation of a complex sugar which is maltose. Now this maltose, it is acted upon by the next class of enzyme, that is the maltase. So maltase, it acts on the maltose to convert it into simple sugar which is the glucose. So that means the starch will give rise to the maltose and maltose will give rise to the glucose at the end. So this will be, the glucose will be the end product of the digestion of the starch. Okay, now the second class of enzymes, these are the proteases. So proteases, they are acting upon the proteins to form the amino acids. So those are the end products. And next class is the lipase. So lipases, they will be acting on fats and they will give us the fatty acids and glycerol. Now if we talk about the chemical digestion of carbohydrates, so we are here taking only the carbohydrate digestion into account. So we start with the salivary glands. So it starts in the mouth. So there we have the salivary amylase in the saliva. It is also called as the ptyalin. So this is our first kind of enzyme that we have in the mouth. So it acts upon the starch or the amylum and converts it into the maltose. So this is the product of this particular reaction. Now next, the pancreas, it is also giving you one amylase which is also called as the pancreatic amylase. So this is also called as amylopsin. So this particular enzyme is also acting on the starch at the duodenum level in the intestine. So that is again giving you the maltose. So the end product of the starch will be maltose, as we have earlier discussed in the previous slide. Now we are coming to the intestinal glands. So this is the third mode of the production of enzymes for carbohydrates. So first we had in the mouth, second we had from the pancreas, and third we have the intestinal glands. So here first of all, the maltose, which was produced as a result of the breakdown of starch, so that maltose has to be broken down. So first of all, this maltose will be acted upon by maltase and that is converted into glucose. So that will be the very first reaction. Then the sucrase, it will be digesting the sucrose and again converting it into the glucose. Same is with the lactose. So all the sugars if we take into account the starch, the maltose, the sucrose and the lactose, they are all digested in different organs at the different level, in the mouth, in the duodenum, in the intestine. So these are all converted into the glucose at the very last. The sucrase, it is also giving us fructose, and the lactase enzyme, it is acting on lactose to give us galactose along with the glucose. Now, if we take up the chemical digestion of proteins, so as you can well see that the protein digestion, it starts in the stomach. So here there is no protein digestion in the case of mouth as such. So it starts with the stomach. So how the proteins they start to degrade? First of all, we have the gastric juices. So these are the juices from the wall of the stomach which are produced by the stomach itself. So here we have earlier discussed that mechanically the stomach also degrades the food into smaller particles, but here we are considering only the chemical digestion. So first of all, we have the gastric juices. Second, we have the HCl, the hydrochloric acid. So that is also produced by the chief cells of the stomach. And next we have got some other digestive enzymes. First of all, it is the pepsin. So that is also produced by the stomach. Then we have the renin. So renin is produced only in the infants, but it stops to be secreted in the case of adults. So here we have the pepsin. So all these enzymes, they are acting on the pepsinogen, the protein or the milk protein, as the case may be. So what is the end product? They may be the polypeptides. Polypeptides means that these are the proteins, but they are smaller in size. So if you have a dipeptide, that means it has got two peptide bonds in between the amino acids. If you have got a polypeptide, so you have many peptide bonds. It is a long chain, and there is a nomenclature which is in between the two. This is called as oligopeptide. Oligo refers to some amino acids or some peptide bonds. So basically from 3 to 9 it is oligopeptide. So if you have dipeptide, then you have oligopeptide up to 9, then you have polypeptide. So these are longer chains of amino acids. But if you see the case of intestine, in this case, the pancreatic and the intestinal juices, they are also containing like trypsin, the enzyme, and we have the peptidases. So these peptidases, they are acting upon the polypeptides to convert them into the amino acids. So finally, at the last, we have got some oligopeptides and we have got amino acids. So that is the final chemical digestion in the case of intestine of the proteins. Now, after proteins, we are taking up the fat. So, in the digestion of the fat, the liver is the most important organ inside our body. So, liver is secreting a particular factor. This is the bile. So bile is stored in the gallbladder. So we all know this, that the bile which is secreted by the liver is stored in the gallbladder. So whenever the gallbladder it contracts, the bile will be released into the duodenum along with the pancreatic duct. So bile is secreted by the time the food is present in the duodenum. So that means the presence of the food in the duodenum, it will trigger the secretion of the bile from the gallbladder, or we can say that the presence of the food in the duodenum will trigger the contractions of the contractions of the gallbladder. One thing has to be noted that the bile has got no enzyme in it. Now, how the bile then acts on the fats if it has got no enzyme? So, how it can affect the fat then? So, there is a process which is called as emulsification. As you can very well see, it has been written over here, emulsification. So, this is the process by which the fat is changed into very small tiny droplets. So this is called the emulsification. So upon emulsification, the fat is changed into small tiny droplets, and those droplets will be further digested by lipases, and that lipase is from two sources. First source is the pancreas, which we called as the pancreatic lipase, and second is the intestine. So we can have lipases from both pancreas as well as the intestine. So they are respectively called as the pancreatic lipase and the intestinal lipase. This is the process of emulsification. So you can very well see, so there is a large oil drop over here. So when it is acted upon the bile, what happens? So we got very small droplets, a number of small droplets of fat. So these fat droplets, they are digested, they are further broken down by the help of the pancreatic and intestinal lipase to the fatty acids and the glycerol. So these are the end products of the fat digestion. So now students, up till here we have discussed the various enzymes which are responsible for the digestion in a typical vertebrate. Now after this, now we are discussing that how the food will travel from the mouth up to the large intestine and how it will be digested. So this is our second part of this particular video. So first of all, we are starting with the mouth. Now how the mouth will help to digest the food partially or mechanically or chemically. So food is chewed in the mouth with the teeth. So we all know that vertebrates, most of the vertebrates, they have got teeth, except like birds, they don't have teeth, they have beaks. So in lieu of this particular condition, they have got gizzards in their alimentary canal. So they are chewing the food in the gizzard rather than in the mouth. But here, if we are taking like a typical vertebrate, so food is chewed in the mouth with the help of the teeth. Now, chewing helps to cut and grind the food into smaller pieces. So this is the mechanical disruption or the mechanical digestion of the food. Now this particular process of mastication, so this is a very important process, the mastication. So it is involving the lower jaw, the mandible. Its lower and upward movement will help to masticate the food. So a combination of these movements, it allows the food particles to be finely ground by the teeth. So this particular process, the mastication or the chewing process inside the mouth, it is also aided by some chemical digestion with the help of the addition of the saliva to the food. So the salivary glands, they are secreting saliva and it is being poured onto the food for chemical digestion. The second thing which is occurring inside the mouth is that while the chewing, the tongue, it rolls the food into small balls. This is a very important fact. So, as the saliva is poured onto the food, it wets up the food. It will help to lubricate the food to go down up to the stomach through the esophagus. So, it will be easier to swallow them. And chemically, it also digests the food. So, the second process after the mastication is the deglutition. So, it is deglutition. Deglutition is the process of the swallowing. So, swallowing that particular ball of the food is deglutition. So, it is accomplished, the process, it is accomplished in 3 stages. So, this is, I am talking about deglutition. The first stage is that the food is brought on the dorsal depressed surface of the tongue by the joint action of the lips and the cheeks. So, we gather the food with the help of the tongue and the upper palate. It is rolled to some extent. Now, the elevation and the backward movement of the tongue, it forces the food into the second part, which is the pharynx. So, food is pushed to some extent into the pharynx from the mouth. So, this is the first stage in deglutition. Now, in the second stage, the food passes from the pharynx into the esophagus. Now, here you can see these are the steps of mechanical digestion. So, the mastication, it is the cutting and chewing of food with the use of teeth. Then saliva is produced, it is poured onto it. The tongue mixes the food with the saliva, it is swallowed. Then we have the deglutition. This is the act of the swallowing. So, the food tube, it churns and mixes the food with the digestive juices in the stomach and the small intestine. This is called as kneading. So, what we were talking about the second stage of deglutition, so that the food, it will enter the esophagus. Now, the pharynx is the common passage for the food and the air, but whenever the food is swallowed, the glottis, it is helping the food to go to the esophagus and not into the respiratory tract. So, this is a very important phenomenon. So, the glottis is helping to direct the food into the esophagus. Now, during this act of swallowing, the inspiratory movements and the expiratory movements, they are suspended. So, you cannot breathe while you are swallowing. So, this is one kind of a risk covering factor, that you cannot respire at that particular time. So, the fluids, they pass rapidly from the pharynx to esophagus. If you are drinking some water along with the food, it will pass directly into the esophagus. This is very obvious. But if you have solid food in your mouth that goes to the pharynx, then it is going to the esophagus. Now, in the third phase, the liquid, it passes directly to the stomach, but the solid, they are forced by the peristaltic movements. This is the third phase. So, as you can see on the right side diagram, that the muscles over the food, they are contracted, but the muscles which are around the food, they are relaxed. So, that means this is a wave of contraction in the esophagus that takes the food down up to the stomach. So, this is the third stage of the deglutition. So, after this process, the food, it reaches the stomach, and I have told you that the stomach has got two parts, First part is the cardiac part, second is the pyloric part. So the cardiac part has got a sphincter which is called as the cardiac sphincter. So when the food it goes to the distal end of the esophagus, it passes through the cardiac sphincter and it goes to the cardiac part of the stomach. Now, the stomach is a muscular bag that lies in the upper part of the abdomen. Now, its muscle can contract and relax, causing the food to break even further. That means the mechanical digestion. And the stomach wall, the inside wall of the stomach has got some constrictions over it. So they are called as rugae, R-U-G-A-E. So these rugae, they are the outgrowths of the inner wall of the stomach, which help in the muscular contractions or the mechanical digestion of the food. Now, the movement of the stomach, the muscular movements of the stomach wall, they help to mix the food with the gastric juice for better digestion. So that means that the wall of the stomach, when it contracts and relaxes, that particular movement is also helping in both the mechanical as well as the chemical digestion of the food. Now, the gastric juices which are secreted by the glands of the stomach wall, they are given into the stomach cavity. So the main enzymes in the stomach, they are proteases, as we have earlier discussed in the chemical digestion inside the stomach. So these are the proteases. Second is the HCl. Now HCl, it gives a particular pH to the food upon which the proteases can act. So here we have clearly written that hydrochloric acid, it helps the proteases to work inside the medium, inside the stomach. So that means that HCl is very much necessary for the action of the proteases. So it is very important to digest the proteins. Whereas the digestion of the fat, it requires more of a like basic kind of environment. So this hydrochloric acid, it also serves the second purpose, that it can kill any kind of bacteria that you have ingested along with the food. So if you have taken some bad kind of food with bacterial growths, so this HCl can kill those bacteria for you. So that is a good thing. So the food stays in the stomach for few hours before passing into the small intestine bit by bit. So as the stomach has got two parts, the cardiac and the pyloric, so the food will enter first of all the cardiac part, then it will be masticated with HCl and proteases, then it will go to the pyloric part, then the pyloric part, it will contract to some extent and passes the food into duodenum through a valve which is called as the pyloric valve. So ultimately the food will be passed to the duodenum or the first part of the intestine. Now, intestine is a muscular tube which is about 6 meters long. In the case of man, it may be smaller or it may be longer in size. The liver and the pancreas, they are connected to the small intestine, but you should remember that these two glands, they are opening via a single duct which is called as the common hepatic and pancreatic duct, which is carrying both the enzymes from the liver and the pancreas to the duodenum part. So the enzymes, they are poured onto the duodenal part. So when the food is coming from the stomach, it is acidic in nature. When it comes to the duodenum, it is acidic in nature. When the bile is poured over that acidic food, it becomes basic in nature, as bile has got the basic you can say it is of basic nature. So it will convert the acidic food into the basic food. Then only the intestinal enzymes can act upon the food. So this is a very, very important point. So as the food passes down into the duodenum or to the jejunum, it becomes basic in nature. The medium altogether changes from acidic to basic in the intestine. So this is because of the addition of the bile to it. Now, food, it mixes with the three fluids in the small intestine to aid digestion. Number one, this is the bile. First of all, I should say bile. So bile is coming from the gallbladder. It is made by the liver, but it is stored in the gallbladder. So when the food it passes to the duodenum, it stimulates the contraction of the gallbladder and secretes the bile from it. Second is the pancreatic juice from the pancreas. So it is coming, it is taking along with it the pancreatic amylase and the pancreatic lipase into it. Next we have the intestinal juices from the wall of the intestine itself. So this juice, it is containing the enzymes like maltase, proteases and lipases. So all these enzymes, they together, they digest the food to the smallest possible fractions that can be absorbed into the blood. So the final digestion and the absorption, it takes place in the small intestine. After the absorption, finally the food enters into the large intestine. So as we have discussed, that large intestine has got two major parts. First is the colon and second is the rectum. So there is a valve again at the connection of the small and the large intestine. Now the large intestine is about like 1.5 meters long. This is in the case of man again. So its function is to absorb water. This is a very, very important function. So you must have gone through it in your previous classes, that it functions to absorb water and mineral salts. So that means it will make your food somewhat dry. So if it is made more dry and dry, so there may be some problem in the passing out of the feces. So you must take an appropriate amount of water along with the food or in between the meals. So that is very, very important. So the function is to absorb water and mineral salts. Now this particular function, it is taking place in the colon region of the large intestine. That is not going on in the rectum. So this is very important thing to note again. Now what is left in the large intestine? It is the solid waste, which is also called as the feces. Now this is a temporary storage, which we call as the rectum. This is the second part of the large intestine. It is a temporary storage place for the solid waste products. Now here it can be stored for some time, and finally that solid waste, it goes out through the anus by a process which is called as egestion. So egestion is the removal of the waste products from the body. So there is again ambiguity. There is a term which is called as excretion, and there is a term which is called as egestion. So there is a big difference between the two. Here we are using egestion as the term. Egestion is the removal of the solid waste products from the alimentary canal, from the anus. Whereas the excretion is the removal of the nitrogenous waste products with the help of the kidneys. So that is the urine. That is a different system. That is a different process. So some students are confused in between these two terms, that what is excretion and what is egestion. So both are altogether different terms and they have got different systems also. Now something about the pancreas. So I'll be taking these slides very quickly as we have taken them before also. So the pancreas, it is producing the alkaline pancreatic juice. So this juice has got amylase, protease and lipase. So pancreas is a very, very important organ in our body. It is producing 3 types of enzymes for you. And moreover, it is also producing some hormones. It is also producing insulin. So the digestion of the food in the small intestine is just like this. So if you are taking like fat, it is digested by the lipase from the pancreas into fatty acid and glycerol. And if we are taking further the starches, that will be digested by amylase or the pancreatic amylase to maltose, and maltose into glucose by pancreatic maltase. So these are very important reactions. So if you are taking up like protein molecules, they are digested by proteases, the pancreatic protease into amino acids. So that's how we can see the importance of the pancreas in your body. Now if we take up the liver, so as we have earlier discussed that it is producing bile. It is stored in the gallbladder. So gallbladder is pouring this bile into the duodenum part. So that helps in the emulsification of the food. So I think students, you are very clear about all the aspects of the digestion of the carbohydrates, the proteins and the fats, and what are the enzymes involved, where are these enzymes formed. If you have a question about the digestion mechanism inside a vertebrate, or you have a question of digestion in man itself, so you have to start from the mouth and you have to go for the egestion, up to the egestion process, with all the enzymes, all the processes which are going on from the mouth up till the formation of the fecal matter. So this particular question is very, very important as per your examinations. You may get a 15 marks full question on this particular topic. So everybody must go by some physiology book. You can go with the like A.K. Barry. You can go with the physiology by Agarwal. You can go with the physiology by Kaushal Kumar. So there are many other physiology books. So you may refer the animal physiology and biochemistry books for this particular thing. I hope that you have got an overall idea about the digestive process. So for this topic, you should be very, very peculiar for all the like processes which are going on in the alimentary canal. Thank you very much.